The interplay between CAR T therapy and antibody engineering, a convergence of immunotherapy innovations, narrated by Luke McLaughlin, biotech digital marketer, business developer, and life science content creator. In recent years, the landscape of cancer treatment has been profoundly reshaped by the emergence of immunotherapy strategies such as Chimeric Antigen Receptor T-cell, CRT, therapy, and antibody engineering. These modalities represent two of the most promising fronts in the fight against cancer, leveraging the body's immune system to recognize and destroy malignant cells. While CAR T therapy involves reprogramming the patient's T cells to target tumor cells, antibody engineering focuses on creating highly specific antibodies that can bind to cancer-specific antigens and recruit immune effector functions. This article explores the interplay between these two technologies, highlighting how they complement each other and could potentially integrate to offer more effective cancer treatment solutions. CAR T therapy, harnessing T cells against cancer. CAR T therapy is a type of treatment in which a patient's T cells are genetically engineered to produce an artificial T cell receptor called a Chimeric Antigen Receptor, CAR. This receptor enables T cells to recognize and attach to specific antigens on the surface of tumor cells. Once these CAR T cells bind to their target, they are activated to kill the tumor cells. The design of CAR constructs is crucial and has evolved through several generations to enhance their effectiveness and safety. The most advanced CARs now incorporate co-stimulatory domains that ensure prolonged survival, enhanced killing ability, and greater overall efficacy of the CAR T cells. T cell isolation and activation. T cells are isolated from the patient's blood through a process called leukapheresis, which separates lymphocytes from other blood components. The isolated T cells are then activated using magnetic beads coated with anti-CD3 and anti-CD28 antibodies, which mimic antigen-presenting cells and provide necessary stimulation signals. This activation induces the expression of activation markers like CD25 and the secretion of cytokines, setting the stage for effective genetic modification. Genetic modification to express CARs. The most commonly used methods for introducing CAR genes into T cells involve viral vectors, such as lentiviruses or retroviruses. These vectors are engineered to be replication deficient and carry the CAR gene construct, which typically includes a scFv linked to transmembrane and intracellular signaling domains. Transduction efficiency is often enhanced by centrifuging viral particles and cells together, spinefection, and culturing them in the presence of cytokines like interleukin-2, IL-2, to promote T cell growth and CAR expression. The genetic modification of T cells to express chimeric antigen receptors, CARs, is a central aspect of CAR T cell therapy, requiring precise biotechnological techniques to achieve. Let's delve into the details of this process, examining the components of CARs and the methods used to integrate them into T cell genomes. Components of a CAR. Extracellular antigen recognition domain. This part of the CAR is derived from a single-chain variable fragment, scFv, of an antibody. The scFv is engineered by linking the variable regions of the heavy VH and light VL chains of an antibody via a short linker peptide. This design allows the scFv to maintain the antigen binding specificity of the original antibody. The choice of scFv is crucial as it determines the specificity of the CAR T cell for a target antigen found on the surface of tumor cells. This means that the CAR T cells will only bind to and attack cells that present this specific antigen, minimizing damage to healthy cells. Hinge region. The hinge region serves as a flexible linker between the scFv and the transmembrane domain. It plays a critical role in allowing the extracellular antigen recognition domain to move and orient itself effectively to interact with the antigens. The length and composition of the hinge can significantly impact the efficacy of the CAR, affecting how the scFv aligns with the antigen and, consequently, the strength of the signal sent to the T cell's interior. Different types of hinge regions, derived from different molecules like CDAA or IgG4, can be used depending on the desired flexibility and immune response. Transmembrane domain. This hydrophobic segment of the CAR anchors the molecule into the lipid bilayer of the T-cell membrane. Stability in the membrane is key for the structural integrity and proper functioning of the CAR. The transmembrane domain typically comes from molecules that are naturally part of the T-cell membrane, such as CD28 or CD3Es, to ensure that the CAR is securely embedded in the cell membrane. Intracellular signaling domains. The intracellular domain of the CAR is critical for transmitting the activation signal to the T-cell's interior machinery once the extracellular domain binds to its target antigen. The most basic intracellular signaling domain in CAR design is the CD3As chain, which is a component of the natural T-cell receptor, TCR complex. This domain contains ITAMs, immunoreceptor tyrosine-based activation motifs, which, when phosphorylated, initiate a cascade of signaling events leading to T-cell activation, proliferation, and the release of cytotoxic agents to kill the cancer cell. Advanced CAR designs often incorporate one or more additional co-stimulatory domains, such as CD28-41BB or ARX40. These domains are included to enhance the T-cell's activation and its functional capabilities. For example, CD28 promotes T-cell survival and enhances cytokine production, while 41BB enhances cellular proliferation and prevents exhaustion, thereby sustaining the T-cell response over a longer period. This highly engineered structure allows CAR T-cells to be potent therapeutic tools, especially in cancer immunotherapy, where they can specifically target and eliminate tumor cells with minimal off-target effects. Genetic modification techniques. Viral vectors. Viral vectors are the most common method for delivering genes into T-cells due to their high efficiency and stable integration into the host genome. These vectors are engineered to be replication incompetent to ensure they cannot cause disease in the host. Lentiviral vectors. Lentiviral vectors are derived from the human immunodeficiency virus, HIV. They have been engineered to remove all viral genes responsible for replication and pathogenicity, retaining only the necessary elements to package the viral vector and deliver the gene of interest. A key feature of lentiviral vectors is their ability to transduce both dividing and non-dividing cells. This is crucial for targeting T-cells at different stages of their cell cycle, ensuring that the CAR gene is effectively delivered. Once inside the host cell, the lentiviral DNA is integrated into the host genome, which allows for the stable and permanent expression of the CAR, which is critical for maintaining the therapeutic effect over time. Retroviral vectors. Retroviral vectors, typically based on the Maloney-Murine leukemia virus, MOM-LV, require cell division for successful integration of their genetic material. This means that they are ideally suited for genetically modifying T-cells, which are actively proliferating during ex vivo culture in the presence of stimulatory cytokines. Like lentiviral vectors, retroviral vectors are engineered to be replication incompetent by removing viral genes necessary for replication and retaining only those needed for packaging and gene delivery. Transfection methods. Electroporation is a physical method used to increase the permeability of the cell membrane using an electrical field. This technique temporarily disrupts the phospholipid bilayer, allowing plasmid DNA or RNA carrying the CAR construct to enter the T-cells. Electroporation. The main advantage of electroporation is its ability to deliver genes without the need for viral vectors, reducing the risk of insertional mutagenesis and other viral-associated risks. However, the expression of CAR introduced by electroporation is often transient, as the genetic material does not integrate into the host genome unless specific mechanisms or sequences are included in the plasmid design, such as those provided by transposon systems. Non-viral methods. These methods offer alternatives to viral vectors by using transposon systems that can stably integrate genetic material into the genome without the use of viruses. Sleeping Beauty Transposon System. The Sleeping Beauty (SB) system utilizes a synthetic DNA transposon and a transposase enzyme. The transposase recognizes specific binding sites, direct repeats, flanking the CAR gene within the transposon DNA construct, Once delivered into the T-cell, the transposase cuts and pastes the CAR gene into the host genome at TA dinucleotide sequences. This system allows for stable expression and has been shown to be effective in clinical applications. Piggyback transposon system. Similar to the Sleeping Beauty system, the piggyback transposon system utilizes a transposase to integrate the CAR gene into the host genome. However, it can integrate larger fragments of DNA and excised without leaving a footprint, which can be advantageous for certain therapeutic applications. Piggyback also integrates preferentially into TTAA sites within the host genome, providing somewhat predictable integration patterns which can be useful for controlling gene expression levels and minimizing disruption of host genes. Each of these methods has its own set of advantages and limitations, which can influence their suitability for specific therapeutic applications. The choice of method depends on factors such as the desired stability of expression, risk of insertional mutagenesis, manufacturing complexity, and regulatory considerations. Ensuring safety and efficacy. The process of genetically modifying T cells to express CARs involves not only the introduction of genetic material, but also rigorous testing to ensure the safety, functionality, and efficacy of the CAR T cells. This includes in vitro testing to confirm that CAR T cells are expressing the CAR and that they can effectively recognize and kill target cells. In vivo studies in animal models to evaluate the therapeutic potential and safety of the CAR T cells. Clinical trials to assess the effectiveness and identify any potential side effects in patients. The continual improvement of genetic modification techniques and CAR designs is critical for enhancing the safety and therapeutic effectiveness of CAR T-cell therapies. Researchers and clinicians work to optimize these elements, hoping to expand the range of cancers treatable with this revolutionary approach. CAR design and construction. The CAR construct design is crucial for the specificity and efficacy of the therapy. It involves creating a fusion protein encoded by a DNA sequence that includes scFv, a single-chain variable fragment. The scFvv is the targeting moiety of the CAR and is derived from monoclonal antibodies. It is engineered by linking the variable heavy, VH, and variable light, VL, chains of an antibody with a short peptide linker, typically about 25 amino acids in length. This configuration allows the scFv to retain the antigen binding specificity of the original antibody. Designing an scFv involves selecting an antibody known to bind specifically to a tumor-associated antigen, and then cloning the VH and VL gene segments. The specificity and affinity of the scFv are crucial as they determine the CAR's ability to recognize and bind to the tumor antigen, driving the subsequent activation of the T-cell. Hinge region. The hinge region connects the scFv to the transmembrane domain. This segment is not merely a passive linker. It provides necessary flexibility and space, allowing the scFv to adequately reach and bind to antigens on the target cells. The hinge affects the orientation and distance of the scFv from the T-cell surface, influencing the CAR's ability to engage with antigen-expressing cells effectively. Hinge regions can be sourced from different proteins, such as CD8A or IgG4, and vary in length and flexibility, impacting the overall function of the CAR construct. Transmembrane domain. This domain anchors the CAR into the T-cell's membrane. It must be robust enough to maintain the structural stability of the CAR on the cell surface under various physiological conditions. The transmembrane domain typically originates from molecules that are part of the T-cell membrane, such as CD28 or CD3Cs. The choice of transmembrane domain can affect the overall expression levels and stability of the CAR on the T-cell surface. Intracellular signaling domains. The intracellular signaling domain is crucial for activating the T-cell following antigen recognition. The primary component is usually the CD3C chain, which includes 3 immunoreceptor tyrosine-based activation motifs, ITAMs. Upon antigen binding, these motifs are phosphorylated, initiating a cascade of signaling events that lead to T-cell activation, proliferation, and cytokine production. Many CAR designs incorporate additional co-stimulatory domains such as CD, 28 or 41BB, CD137. These co-stimulatory domains enhance the amplitude and duration of T-cell responses. CD28 can enhance cytokine production and prevent activation-induced cell death, while 41BB promotes T-cell expansion and longevity. The choice and combination of these domains are critical for the therapeutic success of CAR T-cells, as they modulate the cell's endurance, potency, and overall anti-tumor activity. Expansion and infusion. After the T-cells have been transduced with the CAR construct, they must be expanded in vitro to obtain sufficient numbers for therapy. This expansion is typically done using a combination of cytokines, with interleukin-2, IL-2, being the most common. IL-2 supports T-cell proliferation and survival. This process must be carefully controlled to ensure the CAR T-cells do not become exhausted, which can diminish their efficacy upon reinfusion into the patient. Once an adequate number of CAR T-cells have been expanded, they are harvested, formulated into a therapeutic preparation, and infused back into the patient. The infused CAR T-cells then circulate and, upon encountering their target antigen, activate and proliferate to exert their therapeutic effect, primarily through the recognition and destruction of tumor cells. Each of these steps involves sophisticated biotechnological techniques and a deep understanding of both cellular biology and immunological responses, ensuring the creation of a potent and specific therapeutic agent for cancer treatment. Antibody engineering, precision targeting with monoclonal antibodies, biochemistry and molecular protocols, antibody discovery and development. The generation of monoclonal antibodies typically begins with the immunization of an animal, like a mouse, with a target antigen to elicit an immune response. B cells producing antibodies are then harvested from the spleen and fused with immortalized myeloma cells to create hybridomas that can be cloned. Each clone is tested for antigen specificity and binding affinity. Humanization of antibodies. To reduce immunogenicity in humans, mouse antibodies are often humanized by grafting only the antigen-binding regions, CDRs, onto a human antibody scaffold. This is achieved through recombinant DNA technology, where the genes encoding the CDRs are synthesized and inserted into vectors, containing human antibody frameworks. Antibody engineering for enhanced functionality. Engineering antibodies may involve modifying the Fc crystallizable fragment region to alter their interaction with immune effector cells or the complement system. Techniques such as phage display are used to screen libraries of antibody variants for those with improved binding characteristics or altered effector functions. Converging paths. Synergies between CAR T therapy and antibody engineering. Integrated biochemical techniques. Dual targeting strategies. The development of dual targeting CAR T cells can involve creating tandem CARs, TANCARs, where two different scFvs are linked within a single CAR construct, allowing the T cell to recognize two distinct antigens simultaneously. This is achieved by designing a genetic construct that encodes both scFvs, linked by a flexible peptide spacer. Dual targeting strategies in CAR T-cell and antibody engineering are designed to enhance the specificity, efficacy, and safety of treatments, particularly in the context of cancer therapy. These strategies aim to mitigate issues such as tumor antigen heterogeneity, antigen escape, and on-target off-tumor toxicity. Here I will discuss several approaches used in dual targeting for both CAR T-cells and antibodies. Dual-specific CAR T-cells. Tandem CARs, TANCARs. TANCARs are engineered to express two different scFvs within the same CAR molecule, linked in tandem. This design allows the CAR T-cell to recognize two distinct antigens on the surface of tumor cells. The activation of T-cells requires simultaneous or sequential recognition of both antigens, enhancing specificity and reducing the likelihood of attacking normal cells that express only one of the target antigens. This approach can also overcome tumor heterogeneity where different cells may express different levels of target antigens. Dual CAR T-cells. This strategy involves the engineering of T-cells that express two separate CAR molecules, each targeting a different antigen. This can be achieved by transducing T-cells with two different viral vectors, each carrying a different CAR construct. The advantage here is the flexibility in CAR design and function, as each CAR can have distinct co-stimulatory domains or signaling mechanisms, potentially leading to synergistic effects that enhance T-cell activation and persistence. Split, universal, and programmable. Super CARs. Super CARs use a split architecture where one part of the CAR is fixed on the T-cell surface and the other part, the adapter or switch, is soluble and can be designed to bind different antigens. This system allows for adjustable, reversible, and repeatable control over T-cell activity. By changing the switch molecules, one can redirect the T-cells to different targets as needed, which is useful in managing relapse or antigen loss by tumor cells. Dual targeting antibody engineering. Bispecific T-cell engagers, BiTEs. BiTEs are engineered antibodies that have two different binding domains, one that binds to a tumor-specific antigen and another that binds to the CD3 receptor on T-cells. By engaging both T-cells and tumor cells simultaneously, BiTEs facilitate the direct killing of tumor cells. This dual binding capability enhances the efficacy of T-cell-mediated cytotoxicity even at low antigen densities on tumor cells. Dual affinity retargeting, DART molecules. DART molecules are another form of bispecific antibodies that can engage two different targets. They are constructed from two different antibody fragments, each with specificity to a different antigen. This structure allows them to simultaneously bind to T-cells via CD3 or another co-stimulatory molecule and tumor cells, enhancing the recruitment and activation of T-cells in the tumor microenvironment. Tandem diabodies, TANDABs. TANDABs are a type of multivalent bispecific antibody with two binding sites for each antigen, allowing them to simultaneously bind to two targets with increased avidity. This structure can be designed to engage multiple tumor antigens or one tumor antigen and a T-cell antigen, enhancing the specificity and strength of the immune response. Implementational considerations. The choice of dual targeting strategy often depends on the specific clinical challenge, such as the need to manage antigen escape variants or to reduce toxicity associated with targeting antigens that are also expressed on healthy tissues. Both CAR T-cells and bispecific antibodies must be carefully designed to ensure that their dual targeting capabilities do not compromise their safety, and that they effectively harness the immune system's potential to fight cancer. Each of these approaches involves sophisticated molecular engineering and immunological testing to balance efficacy and safety, addressing the complex dynamics of tumor microenvironments and immune system interactions. Overcoming the tumor microenvironment. The use of engineered antibodies that block inhibitory receptors like PD-1 on T cells or their ligands like PD-L1 on tumor cells can be crucial. These antibodies are typically produced using standard monoclonal antibody generation techniques, but are designed to block interactions that inhibit T-cell function within tumors. Overcoming the hostile tumor microenvironment, TME, is a major challenge in cancer therapy, particularly for immunotherapies like CAR T-cells and antibody-based treatments. The TME is characterized by a variety of immunosuppressive factors, physical barriers, and cellular interactions that can inhibit the efficacy of these therapies. Here, I'll explore several strategies to enhance the functionality of CAR T-cells and antibodies within the TME. Engineering CAR T-cells for enhanced functionality in the TME. Modification for cytokine resistance. The TME often includes high levels of immunosuppressive cytokines like TGF-B and IL-10, which can inhibit T-cell function. To combat this, CRT cells can be genetically modified to express cytokine receptors that convert these normally suppressive signals into stimulatory signals. For instance, a dominant negative TGF-B receptor can be expressed to block the suppressive effects of TGF-B, or a chimeric receptor that converts TGF-B signals into IL-2-like signals can be used to actually boost T-cell activity in the presence of TGF-B. secretion of pro-inflammatory cytokines. Another approach is to engineer CAR T-cells to secrete pro-inflammatory cytokines, such as IL-12 or IL-18, which can reshape the TME by promoting an anti-tumor immune response and activating other immune cells present in the TME, such as natural killer cells and macrophages. This not only enhances the direct anti-tumor activity of the CAR T-cells, but also helps in recruiting and activating the patient's own immune system against the tumor. Engineering for hypoxia resistance. Tumors often have regions of low oxygen, hypoxia, which can impair T-cell function. CAR T-cells can be engineered to express hypoxia-inducible factors, HIFs, that activate under low oxygen conditions. These factors can promote T-cell survival and function in hypoxic conditions, enabling them to be more effective in solid tumor environments. Modifying antibody therapies to overcome TME barriers. Antibody-drug conjugates, ADCs. ADCs are antibodies linked to cytotoxic drugs. The antibody component targets the tumor cells, delivering the cytotoxic drug directly to the tumor site. This can be particularly effective in the TME as it allows for high concentrations of the drug to be delivered directly to the tumor, minimizing effects on healthy cells and overcoming barriers posed by the physical inaccessibility of tumor cells. Bispecific antibodies. As mentioned, bispecific antibodies can engage two different targets. In the context of the TME, one part of the antibody might bind to a tumor antigen, while the other could target a molecule expressed by immune suppressive cells, like regulatory T cells or myeloid-derived suppressor cells, or immune checkpoints such as PD-1 or CTLA-4. This simultaneous targeting can help deactivate suppressive cells or activate effector T cells directly at the site of the tumor. Combination therapies. Combining with checkpoint inhibitors. CAR T cells and antibody therapies can be used in combination with checkpoint inhibitors, drugs that block proteins which prevent T cells from attacking cancer cells, such as PD-1 or CTLA-4. This combination can help overcome one of the major immunosuppressive mechanisms within the TME, potentially leading to improved treatment efficacy. Using oncolytic viruses. Oncolytic viruses selectively infect and kill cancer cells. When used in combination with CAR T cells or antibody therapies, these viruses can help break down the physical structure of tumors, expose tumor antigens, and induce a local inflammatory response that makes the immune environment more conducive to the action of immunotherapies. Addressing metabolic barriers. Tumors often outcompete immune cells for nutrients within the TME, effectively starving them and reducing their functionality. Engineering immune cells to express high-affinity nutrient transporters, or enzymes that allow them to metabolize alternative substrates, can help them sustain their anti-tumor activity under nutrient-deprived conditions. These strategies represent a multifaceted approach to reconfiguring both the biological and physical aspects of the TME. To better support the function of therapeutic agents like CAR T cells and antibodies, each strategy requires careful design and consideration of the unique characteristics of the tumor and its surrounding environment. Universal CAR T cells. The development of universal CAR T cells involves using gene editing tools such as CRISPR/Cas9 to knock out endogenous T-cell receptor TCR genes to prevent graft versus host disease GVHD in allogeneic settings. Simultaneously, CAR constructs designed to recognize broadly expressed tumor antigens are introduced. Universal CAR T cells represent an innovative approach to broaden the applicability and simplify the manufacturing of CAR T-cell therapies. Traditional CAR T-cell therapies are autologous, meaning they are made from a patient's own T-cells, which involves a complex and time-consuming manufacturing process. Universal CAR T-cells, also known as off-the-shelf CAR T-cells, are engineered to be used in any patient, bypassing the need for individualized production. This approach could significantly reduce costs and treatment times for patients. Here's a deeper dive into the technology and strategies behind universal CAR T cells. Allogeneic T cells. Universal CAR T cells are typically derived from allogeneic T cells, which are sourced from healthy donors rather than the patient themselves. This allows for the creation of large batches of CAR T-cells that can be standardized, stored, and used as needed for different patients. However, using allogeneic T-cells presents the challenge of immune rejection and graft versus host disease, GVHD, where the infused T-cells might attack the patient's healthy tissues. Genetic modifications to prevent rejection. To address the issues associated with allogeneic cells, universal CAR T-cells are engineered to minimize their potential to cause immune responses. TCR disruption. The T-cell receptor, TCR, is responsible for recognizing host antigens and can lead to GVHD if the donor T-cells attack the recipient's normal cells. Gene editing technologies like CRISPR/Cas9 or TALENs are used to disrupt the TCR gene in the donor T-cells, thereby reducing the risk of GVHD. HLA knockout. Human leukocyte antigens, HLA, are another component that can trigger immune rejection. By knocking out the genes that express these antigens, particularly the Class I and Class II HLA molecules, the universal CAR T-cells are rendered invisible to the host's immune system, reducing the likelihood of rejection. Incorporation of suicide genes. To further ensure the safety of universal CAR T-cell therapies, these cells can be engineered to include suicide genes that allow them to be selectively destroyed if they cause severe side effects or if GVHD begins to develop. These genes can be triggered by a drug that is harmless to the patient but activates the suicide mechanism in the CAR T-cells. Modular CAR systems. Universal CAR T-cells can also utilize modular systems, such as the previously mentioned SuperCAR system, which uses a universal CAR base that binds to a separately administered adapter molecule. The adapter molecule can be designed to target different antigens, allowing the same CAR T-cell product to be used against different types of cancers by simply changing the adapter. This flexibility could potentially allow one type of CAR T-cell to be used for multiple patients and multiple cancer types, enhancing functionality and safety. Researchers are also working on ways to enhance the functionality and safety of universal CAR T-cells, such as engineering them to express cytokines that promote immune activation or checkpoint inhibitors that enhance their persistence and activity within the immunosuppressive tumor microenvironment. Regulatory and manufacturing considerations. Universal CAR T-cells pose unique challenges in terms of regulatory approval and manufacturing. They must be produced under extremely stringent conditions to prevent contamination and ensure consistency between batches. Regulatory agencies may also require additional testing and monitoring due to the novel nature of these therapies. Universal CAR T-cells are a promising development in the field of cancer immunotherapy, potentially offering faster, more cost-effective treatment options for patients. However, significant technical and regulatory hurdles remain to be overcome before they can be widely adopted. Future perspectives. The ongoing integration of CAR T therapy and antibody engineering is poised to create more potent and precise treatments for cancer. As research progresses, the combination of these strategies may offer solutions to current limitations, such as relapse due to antigen loss, resistance to single-agent therapies, and severe side effects caused by broad immune activation. Conclusion. The interplay between CAR T therapy and antibody engineering exemplifies the potential of combined approaches in biomedical sciences, especially in oncology. By merging the specificity of antibody engineering with the potent effector functions of CAR T-cells, new and more effective immunotherapeutic strategies are being developed. This convergence not only highlights the ingenuity of current scientific research, but also promises a future where cancer can be targeted with unprecedented precision and efficacy. The integration of CAR T therapy with antibody engineering is setting a new frontier in the fight against cancer. This confluence of immunotherapeutic innovations is not just enhancing the precision and power of cancer treatments, but also expanding the horizons of their application. Through the synergistic effects of these two advanced technologies, researchers are overcoming previous limitations and pushing the boundaries of what is possible in oncology. The journey of these combined therapies from bench to bedside exemplifies the dynamic nature of medical research and its potential to yield transformative results. As we continue to explore and refine these approaches, the future holds the promise of more effective, safer, and more universally applicable cancer treatments. The convergence of CAR T therapy and antibody engineering not only marks a significant milestone in cancer therapy, but also lights the path toward a future where cancer may no longer be a formidable foe,